everyone, good morning! For this video, I will show you the step-by-step procedure or process on how to culture bacteria. This is actually a do-it-yourself experiment. Culturing bacteria or microorganisms are being conducted just to know the present bacteria and different samples you have. In my case, I collected my samples in lavatory or sink, refrigerator, money bills, cell phone, and toilet bowl. Microorganisms like bacteria are grown or cultured in a growth medium, for example by using of agar. In order to grow, they need carbohydrates for their energy, mineral ions as well as proteins for their growth, which are present in our medium that we will use. So let's go! First, provide all the things or paraphernalia needed. We have laboratory gown, latex gloves, 1 sachet of agar for our plate, exact amount of water, sterilized plastic containers, which is the alternative petri dish, cotton swab or buds for collecting a sample, alcohol, and tissue. Second, clean and disinfect thoroughly the surface of the table by using tissue and alcohol. Note that we have to wear the laboratory gown and gloves while conducting this experiment. Pardon if I did not use the gloves, but I already disinfect my hand before. Third step, put the agar as our culture media into the water. Stir it well until it dissolved or mixed properly with water. Our fourth step, put the well-mixed agar and water or culture media into the oven, but in my case, I am using here a rice cooker as an alternative since oven is not available. But I surely clean it up and sterilize as well. But then again, much better or advisable to have an oven for heating up the culture media. Then wait for a minute until it boils, or don't forget to stir it. Fifth, prepare the sterilized plastic containers, and then when the agar is now ready, put it into the containers. Remember, just put an exact amount of culture media in each container. After, wait until it becomes solidified. In my case, I waited at least 2 hours. Sixth, after the culture media solidified, collect a sample. Now, I'm gonna collect my sample first in lavatory or sink. Next is in refrigerator. Money bills. Cell phone. And lastly in the toilet bowl. Streak or put the collected sample into the plate or culture media. Don't forget to put a label for us to determine each container. Lastly, place the containers in a warm and dark area, or if you have an incubator, this is much better, and order the bacteria or microorganisms present in each sample to grow. Monitor and observe every day the samples and assess if there are bacterial growth present. That would all the process. The next presentation is the monitoring of our samples from day 1 until to the last day of observation. Thank you!